Our next speaker is Dr. Ulrika Warpman Berglund. And it's another role model that I think is worth paying attention to. So just as a way of introduction here, Ulrika obtained her PhD here in Uppsala in 1997. And then worked, if I understand it correctly, directly into industry.
Yes, I did.
And worked 15 years working in drug discovery and development in several different Swedish-based companies, MSCA, Biovitrum, and Procidian. But somehow, 2012, you got lured over to Thomas Helleday's lab to work in the academic discovery group at SciLifeLab at Campus Solna. Again, a good connection there. We're connecting basic research with the desire to make a practical impact. And there she also served as a principal investigator between 2018 and 2020. She was also a steering board member of the Chemical Biology Consortium Sweden at SciLifeLab for 2 years there. And then she co-founded in 2013 together with, I guess, Thomas Helleday, Oxcia, which is where you are presently. And you've chaired the board there from 2013 to 2021. And you're currently the CEO of Oxcia. And also a board member of Thomas Helleday Foundation. And for those who don't know, Thomas Helleday is also another interesting aspect of how you actually obtain money to fund your research. And he's a very interesting example of that, which maybe you might be thinking about today as we talk. But anyway, the Oxcia is focused basically on modulating oxidative DNA damage and DNA damage response to treat different types of cancers and also fibrosis-related diseases. So specifically targeting two drug targets, MTH1 and OGG1. And I imagine you'll be talking about those.
I will, thank you.
So thank you for being here. So your talk is going to be driving innovation from the lab insight to patient impact. So
Thank you so much. And good afternoon, everyone. Nice to be here and see you. I actually would like to start by asking you a question. What motivates you as a researcher and the ones that are in entrepreneurship as well? Why are you here, I guess, I'm asking. It's not right and wrong, you know. Any answer is correct.
I guess we are hoping to, in my case, hoping to improve health, and in my case, fight antimicrobial resistance.
Right. Yeah. Anyone else?
I would like to understand the world around me.
Ambitious. Good. Anything else?
Instead of having only one application, to broaden the application to more areas.
Yeah. Widen the ideas. Anyone else? You're too shy. Okay. So my motivation started off with the science, curiosity. I wanted to learn, understand how the body works, how the cell works, and particularly when we are sick. And when I did my PhD, I had the benefit to have a supervisor, Professor Agneta Nordberg. She also met the patients. So everything we did, even though it was nitty-gritty details in science, she always connected it to the patient and the patient need. And this is something that I brought with me my whole entire career. But in recent years, since being CEO of the spin-out Oxcia, I also realized a bit what Ulf said in the end, the business case. You need to think about the business case as well. Are there anyone that actually would like to buy your idea? Or the investors that you will get, they also actually would see their investment grow. So these three aspects I think is important. What I will focus on is the science and patient need in this talk. But before showing you how we took an innovation into the patients, I just want to talk a little bit more about Oxcia, which you heard is a spin-out from Karolinska Institutet and Professor Thomas Helleday Laboratory. And we have a mission, and the mission is to make life less short. And we're doing that by our unique technology platform. We call it O2 DDR, where we're targeting oxidative stress, oxidative DNA damage and altered DNA damage responses that can be seen in many unhealthy cells, such as, for instance, cancer and inflammation and fibrosis-related diseases. And the two drugs candidates that we are working with, you can see here, it's OX2-1, which is an oral OG1 inhibitor that we are developing against idiopathic pulmonary fibrosis. And it's in preclinical stage, and we have received an EIC transition grant to partly support this program. And then we have OX101, which is an oral mitotic MTH1 inhibitor. And today we have an ongoing clinical phase 1/2 trial in blood cancer with focus on acute myeloid leukemia. And we have had Vinnova grants for this over the years. We have also seen that this OX101 can work also more broadly. So we have finished an advanced solid tumor phase 1 trial where we are in discussion with Mayo Clinic to do phase 2 in gynecological cancers. And then we have a spin-out program, psoriasis. I will come back to that in the talk. So what I will focus on, I will not mention much of OX2-1, but I will tell you the story about OX101 and the innovation and how we came where we are today. So, cancer, still a leading cause of death. And we believe that the underlying cause to this is that cancer is such a heterogeneous disease. There are a lot of mutations, the cancer cell adapts, resistance mechanisms evolves. And also the existing therapies are very toxic, most of them at least. So what if we could find a drug that actually could work in heterogeneous cancers, in chemotherapy-resistant cancers, and being well tolerated? And this is something that Professor Thomas Helleday was thinking about some time ago. And he came up with a target called MTH1, or NUT1. And at the time, we knew that this protein or enzyme was there in the cell to sanitize oxidized nucleotides from the nucleotide pool, hindering it to be incorporated into DNA and cause damage. And it was also known that if you knock out this enzyme in mice, the mice would grow old and be viable. So it seems like it was a non-essential target. We also knew that the gene is one of the most highly overexpressed genes in cancer versus normal tissue. So the hypothesis was then, well, maybe the cancer cell uses the high level of MTH1 in an oxidative stress environment to make sure that keep the DNA damage so low so the cancer cell is not dying, but can survive it. So our original hypothesis was to inhibit MTH1 so much that a lethal amount of these 8-oxo-DG-TP could be incorporated into DNA and cause DNA damage, and the cancer cell would die. So the team started to look whether this hypothesis was true and did some target validation. And you can see here that when we knocked down MTH1, the cancer cell did not survive. We could also see an elevation of 8-oxo-DG in the DNA according to the mechanism of action. Then we did a human colon cancer cell line that overexpressed a doxycycline-inducible promoter. We took these cell lines, grafted it under the skin in the mice, and let it grow. And then when we had a tumor, we gave doxycycline into the drinking water to deplete the protein. And you can see what happened. The tumor shrank and it disappeared. And we were all excited about it. But at the time, we had no idea whether this target was druggable because nobody had ligands to this more than the substrates. So we also did point mutations in the active site of this protein. Here is one of the point mutations. And you can see, even with this just point mutation in the active site, the cancer cell did not survive. And we could also increase the 8-oxoguanine. So now it was time to actually set up a screening. And we did this together with LCBKI, which is now called CBCS at SciLifeLab. And we set up a biochemical assay, we did several screens, got hits. And me coming from pharma, I really wanted to have a screening funnel. So we set up a screening funnel to optimize the chemistry. We had cell viability to get to see whether we could kill off the cancer cell but still have a therapeutic window. We worked with UDOP here at Uppsala to look at the ADME properties and target engagement in cells. We did selectivity, and then in vivo pharmacokinetics and efficacy in disease models. And for the most interesting compounds, we also set up specific mechanistic studies to look at the atoxo guanine incorporation rescue experiments, the DNA damage responses, how much oxidative stress was important for the efficacy, and tubulin assay, and so forth. So I will stop here a bit before I show you the data, or some of the data, and come back to the team. Because I think team is crucial when you want to do things. And we at Helleday Lab had the possibility to have quite a big team at this time. And you can see here that we had in vitro and vivo pharmacology, we had medical chemistry, we had basic science, and so forth. So with this team, we could actually fulfill quite a lot of things that's needed to go from an idea into the clinic. But of course, we still missed expertises. So, collaboration. We worked a lot with SciLifeLab, CBCS, but also other platforms, DDD platform, Stockholm University, Uppsala University, and Svetox, which is actually key. That does not really exist any longer at SciLifeLab, which I think is a shame. Because what Svetox was it's the toxicology department from AstraZeneca that at the time when they closed down, they actually spin off that department into Karolinska Institutet. Now it is not part of Karolinska Institutet any longer, it belongs to RISE, which is the former SB, process development. And for formulation. And then, of course, the hospital we worked with, and a lot of international collaborations. So with all this network and collaborations, we could fulfill the gaps to actually take this idea into the clinic as an academic group. So back to some of the data then. We did the screens, we got chemical compounds that we start to optimize, the tool compounds, you can see here, we call them 287 and 588. And we also know exactly how they bound into the active site of the molecule. But what we also had, which we think is important, was a negative control. We call it TH650. You can see it's quite similar in structure. There are some differences. And what is important is this is not a very potent compound to this target. So when we did our mechanistic and cellular assays, we could see that our tool compounds indeed selectively killed cancer cells, a number of them, with a decent therapeutic window. And this compound didn't do anything. We also looked at the closest pharmacodynamic marker, the atoxaguanine, and this is a modified COMET assay, where you can see here again that our two compounds could induce the damage in cancer cells, but not in normal cells. So this data and a lot of more data we put together, we published it in Nature 2014. And you can see the number of authors. We were a big team to do this. Before we actually submitted, we had also submitted a patent application on the new chemical entity to secure the patent on the chemistry. Since this publication, we have had almost 40 additional articles on the target, on our compounds, how it works, and the mechanism of action. And as often with new biology and new pharmacology, new things evolves. And today we know that these compounds, they have a dual mechanism. stops the cancer cell in mitosis, causes more oxidative stress, and then causes DNA damage. And more specifically, this is what it does, what we know so far, I should add. We have the compound, we stop microtubule polymerization directly, but also we found new biology that this enzyme is involved in this process. We get more oxidative stress, we get more oxidized nucleotides, we inhibit MTH1 sanitizing efficacy, we get more of those incorporated by polymerases into DNA, and the cancer cell dies. Did you understand it? Yeah?
Sorry for asking one question. I was just wondering, so is this immunosuppressive? Because an adaptive immune response is pretty much,
You would think so, but it is. But we have done immunosuppressive studies, and that it doesn't really in those specific studies. But what we see now in the clinical trials, that the dose-limiting toxicity is on neutrophils. So it does have, depending on some of the... Sorry?
not lymphocytes.
And lymphocytes, but that's really high concentrations. It's mainly neutrophils, actually. But I'll come back to that, because we did see that cancer cells and activated T cells have many similarities with high oxidative stress and elevated levels of MTH1. But it doesn't seem like in the cancer situation it is so important. It's more in autoimmune diseases where we can manipulate the activated T cells. So we don't really fully understand why we see differences. I mentioned in the beginning that we wanted to have a drug that showed very broad anti-cancer efficacy. And this compound do that. So this is a collaboration with Misvik Biology and Juha Rantala in Finland. And he is running ex vivo precision screens. So he takes biopsies, primary biopsies from a patient and put it on a plate and add the compounds. And what you see here is each bar here is one patient biopsy. And you can see that the majority of these patients actually respond to the treatment. Not all, but most of it. When we do some patient-derived ovarian cancer models, this is one from Mayo Clinic, you can also see that we do have really nice efficacy with shrinking of the tumor, or equally good as the standard of care treatment. And we can also combine it. So besides the screening funnel, when I started in Thomas' lab, I also thought it was very important that we knew from the beginning what we wanted to have as a clinical candidate. So preferred and non-preferred properties of the drug candidate. And that is what you see in the table to the right. And we came up, developed the clinical candidate, we call it Karolinska, or TH1579, and now we call it OX101 at Oxcia. And this is a compound with improved potency and PK properties from the tool compounds. It has a good selectivity. It shows a very broad anti-tumor activity. And then it was time to start the safety assessment. And I'm coming back to Svetox, because this was really good for us as an academic group to be able to have the support of a toxicology group that knew exactly what we needed to do. And what we did was to follow a guideline. It's called a ICH-S9, Non-Clinical Evaluation for Anti-Cancer Pharmaceuticals. So if any of you are actually doing drug development in cancer, check this out, because it tells you actually what you need to have before going into clinic. We found the dose where we could start in the clinic. We put effort also, of course, into scaling up the compound and make it into a formulation that the patient could take. And then we put all data in together, IPD, we did the clinical protocol, and sent in a clinical trial application. And it was approved by Medical Product Agency and by Ethics Committee. So we're very happy. And remember, all this we had done as an academic group. So up till now, this was completely academic. Then,
can you say again what SweetTalks is? Is that government?
No, so SweetTalks was a department coming out from AstraZeneca that Karolinska Institutet had for a while. But now it's separated from Karolinska Institutet, and it's in a company called RISE, or an institute called RISE. So we were ready, we're thinking, yes, academic group, we have an innovation, we're now ready to go into the patients. We have all the approvals, but then the leadership at Karolinska Institutet said, no, sorry, we cannot be sponsor for this trial. And there are different reasons for it, but one is that it was a completely novel drug. It was a completely novel target. So they were a bit worried. So of course we were very disappointed, but problems are there to be solved, right? So Thomas Helleday has started a foundation. So the foundation came in and were the sponsor for the trial. And in this instance, we also started to think about, yes, we probably need a company also to be more active in the development of a drug. So Oxcia had been founded a few years earlier, but at this time, Oxcia was also coming in to support the foundation in the sponsorship of this trial. And the first in human phase 1 trial was run by Jeffrey Yaknin, heading the phase 1 unit at Karolinska, and it was called MASTIFF. And there was patient with advanced solid malignant tumors that could be recruited as long as they fulfilled the inclusion-exclusion criteria. For as all phase 1 trials, the aim is to determine safety and tolerability in dose escalation, and then to identify the recommended phase 2 dose and dose regimen. And then as a secondary and exploratory aims, we were looking at signs of efficacy and mechanism of actions. This is a study that now is completed. We know that this is a tolerated oral medicine. We have dose-limiting toxicities with neutropenia, as we talked about, but it's manageable. We have no evidence of chronic accumulate toxicity. And very importantly, we don't see any severe toxicity on renal, kidney, liver, or cardiac toxicity, which is important because many anti-cancer drugs actually causes that. And we did see clinical benefits. We did see some tumor shrinkage, and we did see stable disease up to 18 months in these very, very advanced and sick patients. So what we're doing now is that we are planning phase 2 in gynecological cancers together with Mayo Clinic. They just need the money to be able to do it. Coming back to immune system, as I mentioned, we very early knew that the cancer cell and particularly the activated T cells shares similarities. So when we started this trial, we also had as an exploratory aim that if any of the cancer patients also is suffering from an autoimmune disease, such as, for instance, psoriasis or Crohn's disease, we would capture that. And this is important because if we hadn't had this in the aims of the trial, we were not able to capture it. And then in the end of this trial, we got this lady in. And you can see that she has very severe plaque psoriasis. 11 weeks on our treatment, this is what she looked like. And you can see that she started to heal in her skin. She stopped the trial, she came back 2 months later and was still sustained in the psoriasis. So I was all excited about this. And this is not a serendipity. I mean, the serendipity is that this patient came in, but we have a scientific rationale, we have preclinical data, because we had already published and shown that our mitotic MTH1 inhibitor could actually work in psoriasis, that we did together with Charlotta Enebäck, who is a clinician and researcher at Linköping University. And when I spoke to dermatologists, he said, wow, this is really strong effect. So we were thinking, Oxcia is a small biotech company with 10 people. We are looking at cancer, blood cancer, we're looking at lung fibrosis. We don't manage to also look at psoriasis. So we put all the data together, went out to talk to pharma, and said, this is what we have, are you interested? And last year we actually entered a collaboration with one of the big international dermatology companies. They will develop a topical formulation and take this towards into as a novel treatment for psoriasis. So look upon your data and see. Don't forget, don't be just one way. I guess is my take-home message from this slide. What we have now is a blood cancer trial. It's Stefan Deneberg at Karolinska Hospital who is leading it, but we also have sites in Denmark and Serbia. And we have done the first dose escalation part, and now we are doing a second part with an expansion cohort in acute myeloid leukemia. And why AML? Well, AML is a very aggressive blood cancer. The 5-year survival is less than 20%. And the median overall survival after the frontline therapy is just a couple of months. So there's a very, very high unmet medical need. So that's important. But what is more important for us and our compound is this, that there are accumulating evidence that the high level of oxidative stress promote the induction and development of leukemia in the bone marrow. But then you may think, if you look at this, oxidative stress promotes the disease. What's your instinct? Well, how do you want to treat this? If you have something that causes the disease, what do you want to do? Remove it, right? Yes. And what did I say? How did our OX101 work? We induced more of it. So how does that work? And it's quite interesting because already in the 50s, Donna Farber founder, Professor Farber, actually thought about this. He had children with AML, and he thought, well, let's give them an antioxidant. So he gave them folate, which is an antioxidant. But unfortunately, the children passed away quicker. So then I started to think, maybe I should do the other way around to induce more of it. So he gave an antifolate, methotrexate, and then he could cure the children. So what we are actually doing is building on that concept, causing more oxidative stress, more DNA damage in the cancer cell to kill off the cancer cell, but sparing then the normal cell, hopefully. So why focus on AML? Well, as I mentioned, the mechanism of action in the drug, important in the pathology, yes. We know that ROS-inducible drugs works, yes. We have preclinical support. You see some of them on this slide. We have clinical support. We have partner responder in one of the really tricky AML patients to treat, the P53 mutated ones. And then it's a rare disease, which comes that the market size is not very big. And that doesn't sound too good from a business side, right? But if it's a rare disease, you can get something called orphan drug designation. Has anybody heard about that before? Yeah. And this is quite good, because if you get this, you can get an extension on exclusivity on the market with 7 to 10 years. And OX101, our compound, we just obtained ODD from both FDA and EMA last year. And then, of course, you also need to differentiate what's your unique selling point from other competitors as well. And we think we have it with our compound. I would like to end up by telling you about another collaboration we have. And that is together with Karolinska Institutet and Tom Erkers and his team, where we look at ex vivo precision screens in this ongoing trial. As we didn't really have a biomarker in oncogene saying anyone with oncogene X will respond, we want to go back to the ex vivo precision screen and see, can we actually screen them and say ahead of time you will respond or you will not. So this is a collaboration we have with the hospital Karolinska. And so far we don't have that many patients, so we should be very cautious in conclusions, but I think it looks quite promising. So if we look at patients, we look at this our compound, the green is more responders, pink is non-responders. You can see it's very the patients are very resistant to a lot of compounds, but respond to ours, and particularly where venetoclax, which is one of the standard treatment, are not working. So maybe we have a niche for the venetoclax resistant patient that also our compound can work. Some of my reflections on our journey is, of course, science is important. But next, I think, is a team. The expertises, the mindset, you need to have a champion that really believes in what you're doing, because you will have bumps on the road. And also work with key opinion leaders to help you evaluate your program and also be an ambassador for what you do out in the world. Data. When I came back to academia, I was a bit shocked how the data was not shared at that time. I think sharing data, using electronic lab notebooks, sign off the notebooks, the quality that you can repeat it is extremely important. And then also try to understand what pharma is looking for when you want to meet up. Ask them, is this good enough? Do you need more? Be open to your data. It may not actually be what you think it is. And then if you do drug discovery, you don't need to know everything, but know the process so you know what you're not knowing. And then a bit what you said, Ulf, finance a business case. That's really important. And add it in early and think long term, not just to the next milestone. And for the IP, a strategic plan. I think we did actually a little bit too early here when it comes to patenting. But I do understand why, because we wanted to publish. But it will be a balance, particularly for an academic individual. So, with that, acknowledgement of the team involved. A lot of people, a lot of grants when we were academic and did this. So, if you have any questions, I'm ready to answer. So sharing data and the collaborations, I think MTA. You have a non-disclosure agreement. You have an agreement saying, you know, this is my idea, we work together on this. There could be different options that if you come up with new innovations in the collaboration, that you take a joint IP for that, for instance. I think one of the challenges has been, we touched upon it, that in Sweden we do have this teaching exception, that you as a researcher own your own innovation. In other countries you don't. So most of the time the university are very keen to take IP as well. So that could be a sort of a discussion, but most of the time you can solve it. But always, I think, have something on paper, agreement beforehand, who owns the data, how will you share it, how will you publish it, is one thing. Timelines. I think it depends a lot on money, what you can do. and how fast you can move ahead. I think for us, the first part actually, taking it here to this, from an idea, or actually when we really started an idea to coming to send in a clinical application, I think it's around 5 years. But then the clinical trial as an academic trial has taken a very long time. So if at this time point we actually had Oxcia as a sponsor, we could get much more finance, I think that could have been quicker. But in drug discovery, in companies, you talk about 10 years from idea into clinic, and then could be, depending on what indication you have, it could be anything from 5 to 15, 20 years in clinic. Absolutely, and we did try that. I mean, as I showed you, we see that it works almost everywhere. And I think this is an opportunity, but it's also something that can be a bit challenging. Not when it comes to science and trying to understand, but when it comes to clinical road to get to the market. Because as we did, we had a phase 1 with any solid tumor coming in, as long as you could tick the inclusion-exclusion criteria. But when you look at the data, it's quite a spread of cancers, of tumors. It's very difficult to actually draw conclusions more than on safety. So that's why we now have made a screen, which indication do we believe in most, to focus on that, and then can follow up with other indications that we think also are interested in. Absolutely. We have done a lot of combination studies in vitro and in vivo, mostly in vitro. And what we see is that particularly other compounds that also causes oxidative stress, where we can see more than additive effects. Stubbornness, no. I really believe in the target. I really believe in the compound. At Oxcia also now we do have other programs as well, like the OG1 inhibitor that we also believe in. But I think it's the eagerness to see that your idea actually works in the clinic, and we have so much data supporting that it should and that it is. But I guess you need to be a bit stubborn, because there are days you feel, oh no, not again. I think there have been not really a big doubt, because I think we have so much data showing that this is a target that are important. There have been debates in the literature that MTH1 is not so important for cancer that we have looked at. And I think it's all come down to the environment in the cell and the compounds that we now know that we need to really have oxidative stress in there to see the efficacy that we do. So I wouldn't say, I mean, as a scientist, you always question, right, your data and your science, which we have done all the time. But I think all the evidence that we're generating points to one direction.
One thing in common here, I think both of the speakers today have, I mean, it's the perseverance and doggedly going forward, which is something that is inspirational. But I think it's also illustrative here that, for me anyway, from the outside, the difficulties where you actually look at techniques and technologies that can be broadly applied for analytics versus trying to develop a drug that goes into people. There's totally different things, and I don't know if because there's a huge difference in terms of the technology development we're talking about. So, and I think the challenges of getting a drug that actually works in a clinic and you have long-term side effects and all this other stuff versus actually being able to measure something. So I think I don't want to say from what Ulf has a much easier process of taking your type of technologies into practice versus a drug into practice, right? So
for research, for diagnostics, it's more similar to drugs,
Yeah.
but not as bad as drugs.
yeah, so
Yeah.
that's also very interesting. So,
I have to admit, sometimes I was thinking, why don't I do diagnostics instead? But it's very exciting with drug discovery.
no, it's also very exciting.
Also.
The other thing I want to say is that I know some of you people are in the academic track. We don't think so much about the clinical aspects, and this is also very biomedically, both of these talks. But I think you can also take a look at the basic biology behind it, which is something which I think is really stimulating in terms of looking at oxidative stress and the kind of things. So taking the kind of stuff that's involved in the background of stuff intellectually should be inspirational in that respect. The other thing is that I think some of the challenges for me when I listen to Ulf's talk is that if you're thinking about planetary biology and those kind of more ecological things, confronting many of the challenges that are much, much larger than just creating a cure for a specific patient population that's affecting mankind, climate change and everything else. How do you monitor environments? How do you actually look at the health of different situations? And there we're looking at the type of technologies that Ulf has been bringing out could be actually applied outside of just looking at biomedical stuff. So again, the challenges of thinking about how you use technologies that are derived in some places, thinking broadly, use your own intellect to build on the kind of inspirational models that we've seen today is the challenge to you all. And I'll leave it at that. I really want to thank both of these speakers for fantastic talks and presentations. Thank you.